Hello, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my lecture. So today we're talking about one chapter in human physiology, so that is called locomotion and movement. So welcome to my lecture today. So thank you so much for joining my lecture. So please don't forget to like, share and subscribe. So let your friends know here ma'am is teaching the entire biology for a NEET 2021 aspirant as well as for NEET 2022 aspirants. So don't forget to like, share on your social media. So shall we start off with locomotion and movement, a chapter in human physiology. So hi Natasha, yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So we are done with digestion, absorption. So then we were done with breathing and exchange of gas. Then we went forward with body fluids and circulation, then excretory substances and their elimination. So today we are in locomotion and movement. So we have two more chapters to go in human physiology. After that, human physiology will be done revising. Right, so human physiology, so we'll have complete Menti in human physiology. So that is what is gonna be there. So that is the plan ahead. So we will have unit-wise revision or unit-wise one-shot. So it will be useful for you guys. Yes, Shamita, a very happy good evening. So thank you for joining my lecture. Hi Naveen, hello, how are you? I hope all of you are doing good and your preparation is fine. So there is no confusion. Your concepts are pretty clear. You're just coming to revise in my lecture. So that is how it should be. So please don't start something from scratch which you think is gonna take a lot of time, because yes, your exam is nearing. So just 4 more days in your July, then you have only 1 month, one complete month of August. Okay, so maximum till 15th of August you can still keep studying, but make sure after 15th you only do revision, nothing else at all. Okay, yes, so welcome. So today we're going to talk about locomotion and movement. It's an interesting chapter. So it's been divided into 2 halves. The first half we're going to talk about muscles, the second half we're going to talk about bones. So that is how the entire chapter is. So in bones you will talk about joints as well. So first half is only about muscles. So today is lecture 1. So today we'll talk about muscles, and in the next lecture we'll talk about bones. Yes, Leela, hello, hi, welcome to my lecture. So please do like, share and subscribe. Okay, so here we move, locomotion and movement, lecture 1. So let's go. So this is my schedule for tomorrow. Yesterday we started off with sexual reproduction in flowering plant. So tomorrow we'll continue with the part 2 of this lecture. For all those who are new to my lecture, Monday, Wednesday and Friday I teach a class 12 chapter, and Tuesday, Thursday, Saturday I teach a class 11 chapter. So today is Tuesday, so today we're talking about locomotion and movement. So yesterday we spoke about sexual reproduction in flowering plant, that is lecture 1. So tomorrow we'll continue with the lecture 2. So we'll talk about how pollination happens and what is double fertilization. So that is the topic for tomorrow. So please don't forget to join, because this is also a very important topic. So with that we move forward with locomotion and movement. So I hope all of you are moving right now. Yes. So we are going to talk about right from scratch, what is the difference between locomotion and movement? Are they synonymous terms, or is there a difference between those two? Yes, Antonia B, so welcome to my session. A very happy good evening. Yes, I'm fine. How are you doing? Yes, Shubham sir, hello. A very happy good evening. So thank you so much for joining my lecture. So guys, please do like, share and subscribe. Okay, I hope all of you are moving, everyone is fit, etc. Yes, Shimpi, a very happy good evening. Yes, I'm good, thank you. So yes, Shimpi, so please do like the session. All right, I'm sorry, I'm going back. So everyone is moving, right? Everyone is able to move your hands, your legs, your neck, your tongue, your jaws. Everything is fit. All of you are perfect. All of you are flexible. No issues. Yes, it's dancing like you. Is that so? Yes, so perfect. No problems in your bones, your muscles and joints. Shall we check? How many movements can you do? Not dance movement? Yes? Okay, so Anthony says locomotion is different from movement. Okay, so let's check it out. So how many types of movements can you do? You can move your limbs, you can move your legs. Then yes, you can of course move your shoulders. You can move your wrists, so that is also a movement. You can move your jaws. right? You can move your tongue. So these are the different types of movements. Ma'am, we can move even our eyelids. Okay, fantastic. Fine. So at least you know what kind of movements you have. Okay, so with this introduction, let's move forward. So this is how the chapter is divided. So these are the topics that we're going to talk about. So we're going to talk about types of movement. We're going to talk about what is a muscle. So what are the types of movement we have? So before that, we're going to define what is locomotion and we're going to define what is movement. So then we talk about the different types of movement in your body. Yes. So then we'll talk about muscles, the types of muscles which we already have an idea of. Then we move to skeletal muscle. How is a muscle designed, or what is the architecture of a muscle? So how intricately it is designed, and when I move my hands like this, so what is the movement of my muscle, right? So all that is important. So then we're going to talk about what are joints. So then disorders of a muscular and skeletal. Okay, so this is muscle and this is skeletal system. So here we are going to talk about bones. So what are the different types of bones that we have in your body? So how many bones you have? Yes. So followed by joints. How many joints you have? Yes, ankle movement, wrist movement, knee movement. So movement of your spinal cord. So you have so many type of joints. So joints is which connects two bones, right? So we're going to talk about them, and finally we'll end the chapter in disorders of muscular and skeletal system. Okay. How many movements are there? Okay, so we're going to talk about them. Yes, Shimpi, ready? Okay, so here we move forward. So first let's talk about what is locomotion. Yes, we have 206 bones. So you're done counting? Yes, everyone count and see if you have 206 bones and tell me. Ma'am, it's not possible to count. Don't worry, we'll count in the next lecture. First, let's now concentrate on muscles, okay? So locomotion, so let's define what is locomotion. So before we talk about locomotion, so let's talk about movement. It is a significant feature of living beings. So be it bacteria, be it plants, be it animals, all living organisms move. Ma'am, plants, how do they move? There are certain plants which moves, right? Like your climbers, etc. But movement is common in living organisms. Living of plants, your animals, your microorganisms, your humans, we all move, right? So it is a significant feature which is classified as living. So streaming of protoplasm in amoeba is a simple form of movement. So amoeba is a protozoan, right? So this how does it move? It is completely ciliated. So the movement of cilia pushes the entire protoplast. So that is also a movement, right? So now inside your body, food is pushed down from your esophagus into your stomach, from your stomach into your intestine. So that is how it moves, right? So how many bones in a baby? 306 in babies. Are you right? Do we have 300 bones in babies and 206 in human adults? Yes, find out if your answer is right. Huh? Okay, so movement of cilia, flagella, tentacles. So if you remember animal kingdom, we spoke about hydra. So hydra comes under phylum Coelenterata. So it has long tentacles, right? So they also show movement. Then what is the difference between movement and locomotion? So this is my bacteria. So bacteria has flagella and the small hairy structures are my cilia, right? So in one way or the other, it's gonna show movement. So this is my hydra, so this has tentacles, so these are known as tentacles. Okay, so human beings can also move, so we move our limbs, our jaws, our eyelids and our tongue, right? Okay, so the movement which results in a change of place or location, which is in turn a voluntary movement, is known as locomotion. So movement is a broad term, right? So moving my muscles is also movement, moving my eyelids is also movement, but the one which results in the change of location or direction, or which is a voluntary movement, is locomotion. So if I want to stretch my hand, yes, it is my voluntary movement, right? So I am willing to do it. So my brain is taking my command and the brain is in turn giving me command, right? So this is how it is. So anything that results in the voluntary change of movement or voluntary movement which results in the change in direction is known as locomotion. All right, fine. So moving further, so that is the difference between locomotion and movement. So don't get confused. So movement is a broad term. So one type of movement is locomotion. So locomotion results in the change of location. Right? So it is a type of voluntary movement. Right? Now, the next question is, movement and locomotion, do they have different structures? Do they have different structures? Yes, Munna, hello, hi, a very happy good evening. So thank you so much for joining my lecture. So I hope you're doing good. So please do like, share and subscribe. So 300, okay. You checked it in Google, so Google said 300. Okay, yes, they have a bit more bones because the bones are not joined. So someone is here, Haji Muhammad, yes, welcome. So he says NEET is postponed. Is that so? So you're getting flash news more quickly. Well, we are not that informed. So till the time I came inside the studio for class, there was no such news. Okay, so yes, Leela, you're right. So it is all locomotions are movement. All locomotions are movement, but all movements are not locomotion. Right? So please remember. So now how about the structure of your movement and locomotion? So movement is of any type. Locomotion is the one which results in the change in direction. For example, I move my eyelid. Right? So this is not locomotion, but this is movement, right? Moving my eyelid do not result in a change of my location or direction, right? Movement of my neck. So what it is? Is it a movement or locomotion? Yes. So do they have different structures or the same structure? So that is the next question in line. So here we go. So locomotory structures need not be different from those affecting other types of movement. The structure can be same, which can result in movement as well as in locomotion. So example, in paramecium, cilia helps in the movement of food through cytopharynx and locomotion. So paramecium has cilia which helps in the movement of food. So it also helps in the movement of the entire cytoplasm as well. So it also helps in locomotion. So this is your paramecium. So it is a ciliated structure. So the cilia helps in the movement of food. So it helps in the movement of food as well as it helps in the movement of the entire organism. So this is paramecium. Okay. Ma'am, Didi ko maro bahut acha. Wow. Why? Shimpi is a good girl. She is listening to class quietly. Why? Okay, so Hydra uses its tentacle for capturing its prey, and it also uses the same for locomotion as well. So this is used as a normal locomotory structure as well as a structure for movement. Thus the structure for locomotion and movement can be same. So what are the different types of locomotions, or what are the different types of movements that we have? So before we go, so this is what we discussed. All locomotions are movement. All movements are not locomotions. So the method of locomotion is performed by animals and it varies within their habitats and demand of solution. So if you see a dog roams around so much in a street, right? But the cow does not roam around much. It just sits down and chews. It just does cut chewing, right? So the need depends upon the demand of the situation as well. So why do an organism locomote? So what is the necessity? What is the need? So here, in search of food, yes, a dog moves around in search of food, right? Then shelter, yes, now it's raining in Kota, so all the dogs try to find shelter so that they can sleep peacefully, so that the trees do not fall, there is no sound, there is no water, etc. And the next one is they find a mate for themselves and suitable breeding grounds that has favorable conditions to them, climatic conditions, and escape from enemies. Of course, the enemies for dogs these days, stray dogs, have become humans. So this is the reason why they locomote. Yes, ma'am, welcome to my lecture. So very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Okay. Oh, she hits you. Okay, let's see if she disturbs you in class. Then I will scold her. Okay. Fine. So this is about locomotion and movement. So now we're going to talk about the different types of movement. So it is raining there also. That's really nice. Why are you sad? So rain actually makes you feel better than all the heat. So it's been raining in Kota since last night, and yes, it has reduced a bit now. So it was raining till 4 o'clock, but after that it reduced, but before that it was raining very heavily. Okay. So types of movement, so we have 3 types of movement. The cells of the human body, so we are talking about the movement in human body. Shouldn't answering your question. So the cells of the human body exhibit 3 main types of movement. One is amoeboid, ciliary and muscular. Okay. So that is forage of food. So F-O-R-A-G-E. So forage is also in search of. So that is an equivalent term. Okay, Antonia B? Yes. So cells of the human body, they exhibit 3 types of movement. One is amoeboid, ciliary and muscular. So amoeboid movement is exhibited by some specialized cells in your blood. So in blood we have macrophages and leukocytes. So macrophages is also a type of immune cell that acts as a guard cell. So macrophages also present your antigen to your T lymphocyte. So some specialized cells in the blood like your macrophages and leukocytes, they exhibit amoeboid movements. So next, cytoskeletal elements. So we have microfilaments. If you remember cell, the unit of life, we were talking about cytoskeletal elements. So cytoskeletal elements include your microfilaments and microtubules. So we spoke about actin and tubulin there. So those also show amoeboid movement. The next one is ciliary movement. They are internal but they are present in tubular organs. So we know columnar or cuboidal ciliated epithelial cells. They are present in the bronchioles and in your fallopian tube, right? So that is ciliary movements. Example, trachea and the passage of ova, right? So the movement of our limbs, jaws, tongue, they require muscular movement. So that is the 3rd type. So this is muscular movement. Okay. Yes, Dharni, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So locomotion needs the perfect coordination activity of muscular, skeletal and the neural system. So this chapter talks about muscular and skeletal system. Okay. So this is your ciliated movement. Okay, so before we go into muscles, so do you remember the examples of the movement? Yes? Shall we check? So we are talking about movement, right? So we have 3 types of movement. So what are the 3 types of movement we just spoke about? So we have amoeboid movement. So who shows amoeboid movement? Examples, cells of your blood. So cells of your blood include, what are the cells? So this includes macrophages. So this includes leukocytes. They show amoeboid movement, right? So the next one, who is? It is your cytoskeletal elements. So what makes up your cytoskeleton? Your microfilaments and microtubules. So this is made up of tubulins and this is made up of actin. So all these also show amoeboid movement. Okay. So then the next one is your ciliary movement. So ciliary movement is shown by your trachea or your bronchioles. Then you have the fallopian tube. So where you have the passage of ova. And the last type of movement. So what is the last type of movement? Ciliary, amoeboid and muscular. So muscular movement includes all your limbs, your tongue, etc. Yes, you have what is flagellary movements that is not present in your humans, that is in your bacteria. Okay, so these are the different types of movement. So now let's move forward and talk about the structure of a muscle. Oh, I'm sorry, I'm going backward. So now let's talk about the structure of your muscle. So welcome to muscle. So muscle is a specialized organ of mesodermal origin, right? So muscle develops from the mesodermal layer. So 40 to 50% of the body weight is owned by muscles. So muscles have specialized property like excitability, contractibility, extensibility and elasticity. You know what is contraction, you know what is elasticity, right? So what is excitability? Yes, it can even transmit or conduct an impulse, or it can have action potential. So that is known as excitability. Okay, so muscles are classified based on the location, the appearance and the nature of regulation. So they are based on the location. So based on the location, you have three types of muscles. The first one is skeletal muscle, then you have visceral, and the third one is cardiac. Okay, yes, Ayyappan Shaktivel, a very happy good evening. Yes, I am good. How are you? So thank you for joining here. So please do like, share and subscribe. Yes, I missed flagella movement. So what is the example of flagellary movements? So which cell in your body shows flagellary movements? Yes, give me the example. So is flagellary movement showed by humans? Yes, neurons can excite. So muscles to some extent they are also excitable. Okay. So we have three types of muscles. So muscles originate from the mesodermal region, and 40 to 45% contribution is by muscles in your body weight. So they have special properties, 4 properties. What is it? So it is EEC. Excitability, extensibility, elasticity and contractibility. So based on the location, appearance, they are divided into 3 types: skeletal, visceral and your cardiac muscles. Okay. Neurons, okay, so sperms show flagellary movements. Yes, right. So trypanosoma, that is protozoa, it is a protozoan, so that also shows flagellary movement. Yes, perfectly right. Yes, Swami, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So we spoke about ciliary, amoeboid, muscular. So we also have flagellary type of movement. In humans, it is shown by sperms, and in terms of protozoans, it is shown by flagellated trypanosoma. All right. Okay. So we have 3 types: skeletal muscles. So skeletal muscles are striated, meaning they have stripes. Skeletal muscles, can you see it has stripes? So it is striated, and yes, it is attached to your skeletal system, and it is voluntary. Yes, of course, my hands have bones. Yes, it is attached to my skeletal system, but voluntary movement. I can move whenever I want, right? So that is your skeletal movement. The next one is visceral. So visceral is also known as smooth muscles. So this is non-striated. Okay, and this is involuntary. So this is not under your control. So this is smooth muscles. This is present towards the hollow organs in your body. The third one is cardiac. So this is associated with the heart, and this is also striated. So this also has stripes, and this is also involuntary. So these are the 3 types of muscles that we have. Okay? Yes. So this is smooth. So this is cardiac. So these 2 are striated. So these 2 are involuntary. Fine? Okay. Done. So now we move forward. So let's talk about the structure of a skeletal muscle. So how is a skeletal muscle present? So we are talking about a skeletal muscle. We are not talking about smooth or cardiac muscle, because skeletal muscle is voluntary, right? So it is easy for us to understand. So we go forward with your skeletal muscle. Okay. So your muscle is like this. Okay. So this is your skeletal muscle. So inside the skeletal muscle, you have bundles like this. Okay, so these are known as muscle bundles, or it is known as fascicles. Fine. So in between two muscle bundles, I have a connective tissue. So this connective tissue which supports two muscle bundle, so it is a collagenous connective tissue which is known as fascia. Okay, yes, CIA. Okay, so what is present inside these muscle bundles? So here I draw the muscle bundle like this. So I am enlarging this. So inside the muscle bundle, I further have something else. So this is my muscle bundle. So inside this, what do I have? I have muscle fibers. So these two are my muscle fibers. Okay, yes. So inside my muscle fiber, I have what is known as myofibrils. So inside the muscle fibers, I have myofibrils. Okay, so muscle fibers, so I am talking about muscle fibers. So muscle fibers have a type of plasma membrane. So that is known as sarcolemma. Okay, so they have a cytoplasm which is known as sarcoplasm. They have an endoplasmic reticulum which is known as sarcoplasmic reticulum. Fine. So this is the architecture of a skeletal muscle. So skeletal muscle has muscle bundles which is also known as fascicles. So they are joined together by a collagenous connective tissue that is known as fascia. So inside the muscle bundles, I have muscle fibers. So within the muscle fibers, I have myofibrils. Okay, so the muscle fibers can have a plasma membrane and it is called a sarcolemma. It can have a cytoplasm which is called a sarcoplasm. It can have an endoplasmic reticulum that is known as sarcoplasmic reticulum. Okay, yes, fine. So now we move forward. So each skeletal muscle is made up of number of muscle bundle called as fascicles and they are held together by a connective tissue. So within the muscle bundle, I have muscle fibers. So here they have a plasma membrane which is sarcolemma which encloses the sarcoplasm. So the muscle fiber has many nucleus. It is coenocytic condition. Muscle fiber is inside the muscle bundle, so that has multi-nucleus condition, that is coenocytic or synticum. So synticum is multi-nucleate condition. So they have an endoplasmic reticulum, that is sarcoplasmic reticulum. So look here, this entire thing is my skeletal muscle. So I have bundles like this, they are my muscle bundles. So they are connected to each other by fascia. So inside each of the bundle, I have fibers. So these are my muscle fibers. Okay. So inside my muscle fibers, I have my myofibrils. Okay. So it is how it is. So I have a muscle bundle. Okay, muscle. So muscle bundle, muscle fiber, inside that I have my myofibrils. So my myofibrils are striated. So once again watch it from start so it will be easy to understand. So muscle, muscle bundle, muscle fiber, inside that I have myofilaments or myofibrils which has striation. Okay, is this clear to everyone? Understood the outside architecture? No doubts? Okay, so now let's move further. Fine. So we are going to talk about light and dark bands. So I told you we have what is known as myofilaments or myofibrils, right? So these myofibrils have, so inside the muscle fiber, you have a number of parallelly arranged filaments in the sarcoplasm that is known as myofilaments or myofibrils. So they are synonymous terms. Myofilaments and myofibrils are the same. They are present inside your muscle fiber. So each myofibril has an alternate dark and a light band. Okay, so the dark band is because of a protein known as myosin and the light band is because of a protein known as actin. So I have checks, right? I have striations. So I have alternate thin line and dark line. So it is the normal meaning. So here I have a thin line. So then I draw a thick line like this. Then I draw a thin line, then a thick line like this. So this is light, dark, light, dark. This is how it is. So the light color is because of a protein known as actin and the dark color is because of a protein known as myosin. So actin protein causes the light band. The light band is also known as I band or isotropic band. Iso is same. It is very light and it is same everywhere. So actin gives you light band which is also known as I or isotropic band. So myosin gives you a dark band which is called as A or an isotropic band. Okay, so this is how it is. So this is how it is. You have a light band, you have a dark band. You have a light band, you have a dark band. Light band and then you have a dark band. Right? So we know the light band is I band. Okay. So the I bands are joined together by what is known as a Z line. So if you look, this is like Z, right? So the I bands, that is the light bands, are joined together by Z line. So this is your Z line. Okay. Can you see? This is your Z line. Fine. So the line that is in between your A band. Can you see? This is your dark band. So dark bands are A band. So this portion of A band is not overlapped by your I bands. So this portion is known as your H zone. Okay. So your dark bands are held together by what is known as an M line. Okay. So the successive distance between two Z line is your sarcomere. So don't worry, I will explain if you have not understood. So we have light band and dark band, right? So these are my light bands, okay? So now I have a dark band. Hi Satyendra, after a very long time you are here. Yes, very happy. Good evening. Yes, I'm good. Thank you. How are you? I hope you're doing good. So thank you for joining a session. So please do like, share and subscribe. Okay. So here I have a dark band. So this is the middle of my dark band. Okay. So now I will do all the markings in blue color. So this is my light band. So light band is also known as I band, right? So this is my dark band. So dark band is also known as A band, right? So I bands are joined together by what is known as Z line. So Z line holds the I bands together or attaches or holds I bands together. Fine? Okay. Now, here your dark bands are held together by what is known as M line. So, this is your M line. So, the portion of your A band that is not overlapped by I band. Can you see? This is the portion of A band that is not overlapped by I band. So, this is known as H zone. So, this is also your Z line. So, the region between two successive Z line is known as a sarcomere. So sarcomere is the region between two successive Z lines. Okay, so this is the sarcomere. So sarcomere is the structural and functional unit of a muscle. Yes, okay. So you understood all the terms that is on the board? Yes, any doubts? You understood all the terms that is on the board? Any doubt? Is this clear? You understood what is a Z line, M line, head zone and what is a sarcomere? Yes, all of you? I hope there are no doubts. So can I move forward? Till this it's clear? Yes. So here dark band is made up of what is known as myosin protein and light band is made up of your actin protein. So now let's talk about the structure of actin and myosin. Okay? Yes. So here we move forward. So the same diagram I have replicated. So structure of contractile proteins. So we have two contractile proteins. One is actin, one is myosin. So every time I put my hands like this and fold, yes, my contractile proteins are working. So who are my contractile proteins? Actin and myosin. Okay? Because sarcomere has your actin and myosin, right? So when you move your muscle, it is your sarcomere that performs the function. So in kidney, your nephrons are the structural and functional unit because that filters, right? So in muscle, sarcomere is the structural and functional unit because, so when I move it and put it like this, it is my sarcomere that is showing some kind of activity. Okay, so now let's see the structure of the contractile proteins, actin and myosin. Okay, so each actin filament has two F filamentous actins. So we are talking about actin. Okay, so actin is responsible for the light band, right? And it is also known as the I band or isotropic band. So what is the structure of actin? So each actin filament, so you have many I bands, right? So each actin filament has two F filamentous actins helically wound around each other. So it is somewhat like this. So this is the structure, okay? So it has two F actins. So can you see the yellow color balls, yellow color circles? Yes. So this yellow color is double stranded. Okay, like your DNA. So this each one is a F actin. So it is ultimately like this. So each one is a F actin. So this is one F actin, that is filamentous actin. So this is another filamentous actin. Fine? Okay. So each F actin is a polymer of G actins. So this is one F strand and this is one F strand. So just like we say cellulose is a polymer of glucose. So this is one example I am giving. So cellulose is a polysaccharide. So what are the individual units here? Glucose. Right? Repeating structure of glucose makes up cellulose. So glucose is the monosaccharide. Yes, these two are filamentous actin, but each circle monomeric is a globulin, is a globular actin. So globular actins make up filamentous actins here. So each round ball is a G actin which together makes up the F actins. Okay. So apart from this, very close to the structure, another protein also runs. That is known as tropomyosin. So this is like this. So very close to this, I have another protein also running. So this is my tropomyosin. So this brown one is my tropomyosin. Okay. So it has a complex protein on its top. Can you see the brown color is my tropomyosin, but at intervals in your tropomyosin, I have this three dot shaped structures or three flower shaped structures, right? So on regular intervals on my tropomyosin, I have what is known as troponin. right? What is on my tropomyosin? It is troponin. Okay, so actually your actin and myosin has to bind. So if I have a muscle movement, my actin and my myosin has to bind. Okay, so where will they bind? At this regions of tropomyosin. So now when troponin is bound to tropomyosin, actin and myosin will not bind. The moment troponin moves away, then myosin will attach to tropomyosin and then you have muscle contraction. So this is the structure of actin. So we are talking about the structure of contractile proteins. Okay, so first one is actin. So actin has two helical structures of F-actin. Individually, it is made up of G actins. Very close by, it has another filamentous protein, that is tropomyosin. So on tropomyosin, we have troponin. So your actin and your myosin binds for an efficient muscle contraction. Okay, so this efficient muscle contraction, so your myosin binds to actin at this site. The moment troponin moves away from this, your myosin binds here. So this is how the muscle contracts. Okay, fine. So now I move further. Now we are talking about the structure of myosin. So this is my structure of myosin. So pay attention, it is a polymerized protein. So the structure of myosin is divided into two parts. One is the head part and the other one is the tail part, because it has a head and a tail. Okay, so the head part is very heavy. So it is known as heavy meromyosin. So myosin is a polymerized protein. So the individual units are meromyosin. Okay, so this entire thing is a polymerized protein. So individual units are known as meromyosins. So this is my heavy meromyosin. The head portion with the neck is a heavy meromyosin structure and the tail portion is a light meromyosin structure. Okay, yes. So your former, that is the head, is heavy meromyosin. The later is light meromyosin. If you look at the structure of the head, so it has ATP binding sites and it also has binding sites for actin. So this is where the actin, the tropomyosin portion will bind, and it also has ATP binding sites. Okay, so this is the structure of your myosin. Fine. So now how does it happen? So now we have actin, right? So actin is somewhat like this. So this is my F-actins. So very close by, I have my tropomyosin as well. So on tropomyosin at regular intervals, I have troponin. So this is my tropomyosin. So this is my troponin. Right? So we know the endoplasmic reticulum of your muscles is sarcoplasmic reticulum. So this has high concentration of calcium ions. So this high concentration of calcium ions causes the troponin to bind here. So when the calcium concentration in your so your sarcoplasmic reticulum has high calcium ions. Okay, so now this moves into the sarcoplasm, this moves into the cytoplasm. From the endoplasmic reticulum of the muscle, it moves into the cytoplasm. Okay, so now your cytoplasm has increased calcium ion concentration. To counteract this, troponin goes and binds to the calcium. Now the space is free for the myosin to bind, right? Because where troponin is there, that is where your myosin is going to bind, right? So your myosin has a structure like this. Right? So here it has an actin binding site. And it also has an ATP binding site. plus it also has an ATPase activity. The moment troponin moves from here and binds to the calcium, and ATP binds to the head of your myosin, and the ATP is processed by the head portion. So if ATP binds here, ATP is converted into ADP plus PI. The moment ATP binds to your myosin, there is a conformational change such that your actin and your myosin will bind. The binding of actin and myosin is such that a cross bridge is formed. The moment ADP plus PI comes out, actin and myosin connection is broken out. So that is how it happens. So this is known as sliding filament theory. Okay, so how it happens? Okay, so this I will explain, but before that let's talk here. So muscle contraction. So see, this is my myosin filament, the head part. This is my actin filament, right? So now when my actin and myosin bind, so this is cross bridge formation. The moment ATP is hydrolyzed, so this happens when ATP binds. When ATP comes out as ADP plus PI, the cross bridge is broken. So this is how your muscles contract. So why does it begin? Because your sarcoplasmic calcium concentration is pretty high. So what increases this sarcoplasmic concentration? That I will explain here. So pay attention. So who brings the signal for your muscle movement? Your neurons. So neurons which come from the CNS are known as motor neurons. So motor neurons bring signal from the brain, right? Yes, so this brings the signal from the brain into the muscle. So what is the structural unit of a muscle? It is your sarcomere. So here I am talking about a muscle fiber to be specific, right? So motor neurons bring a signal here. So this junction between a neuron and a muscle is known as neuromuscular junction. So this is the neuromuscular junction. So this motor and this muscles, okay, so they together form the motor end plate or the motor unit. Okay, because this is the motor, this is the neurons and this is the muscles, right? So this junction is my neuromuscular junction. So we know this transmits neurotransmitter, this releases neurotransmitters. So when the neuron which is bringing signal from the CNS, it transmits or it releases the neurotransmitters here. The neurotransmitters activate the muscle fiber. So it activates the muscle fiber such that the calcium ions from the sarcoplasmic reticulum enters into the sarcoplasm. So this increased calcium ion causes troponin to bind. Thus, it gives space for actin and myosin. Clear everyone? Yes, Sriram. Hello. Yes, Grace. Hello. Welcome to my lecture. So, thank you for joining. So, please do like, share and subscribe. Is this clear to everyone? Yes. Yes, you understood. So, this is your muscle movement. So, here I have a GIF. So, you can see. So, now troponin moves. So that your myosin binds. So that space is where your troponin is binding. It's not exactly troponin. Troponin has moved. So that is why your myosin is binding. So this is cross bridge formation. Yes, clear everyone? Yes? Yes, the neurotransmitter is acetylcholine. You're right. Yes, fine everyone. So did you understand the first part of this lecture? Yes? No doubt? Okay, so we will talk about red and white fibers. So in red fibers, so we are talking about muscle fibers. which is red in color. That is because of a pigment called as myoglobin. So when this pigment is there, it has high amount of mitochondria then it is used in the formation of ATP. So they are mostly aerobic muscles. You have two types of muscle fibers, red and white. So red is because it has a pigment known as myoglobin. So this contains mitochondria, thus used in the synthesis of ATP, and it has aerobic muscles. The next one is white fibers. The white fibers have less of mitochondria, but they have more of sarcoplasmic reticulum. Thus, they have increased calcium ion concentration. So it is anaerobic process. Okay. So when your muscle movement happens, when your actin and your myosin binds, the actin overlaps your A band. The I band overlaps your A band. So now the length of I band shortens, but the length of A band remains unchanged. So that is the logic. So please go through your textbook so that it's clear once again. So once you read the textbook and you look into my lecture for revision, it is perfect. That is done. Okay. Yes, Kalpata, a very happy good evening. So thank you for joining my lecture. So please go through the lecture and see if you have any doubts. So with this, we are done with the first part of the lecture. So that is your muscle movement. So sarcomere is the structural and functional unit of muscle, and it is the region between two successive Z lines. Z lines hold the I bands together. Okay. Yes. So with this, we come to the end of this chapter. So please join me tomorrow at 5 p.m. So we are going to continue with sexual reproduction in flowering plants. We will continue locomotion and movement on Thursday. Okay. So if you genuinely liked my way of teaching, so please do like, share and subscribe. So this is me wishing you more and more. So take care, stay healthy, stay tuned. I will meet you tomorrow. So until then, bye and take care.